Hello lovelies, in this video we're going to be going through the paper that we've written for 2024 for your Edexcel IGCSE Biology Paper 1. So if you haven't had a go at this paper already, what we will need to do is to pause after the question comes up, have a go at it yourself on a bit of paper and then play and we can work through the answers together. And we'll show you what sort of things the examiner will be looking for, we'll show you what sort of things the mark scheme might be pointing to. We will help you structure your answers for your exam.
Different groups of organisms have different features. Complete the table by ticking features that are present in each group of organisms. So this is about kind of classification groups, what each organism has as part of its kind of features that allow you to decide which group it should be in out of plants, animals, fungi and prototists. And remember, these are the different kingdoms. So it says to only tick if they do have them. So if this is like a defining feature of this kingdom of organisms, then we're going to tick it. So do they have cell walls, like around their cells? Do they store their carbohydrates as glycogen, not starch? Starch is only stored in plants. And are they all single-celled organisms? So are they like bacteria, for example? So do plants have cell walls? Yes, they do. Do animals have cell walls around their cells? No, they don't. Do fungi? Yes, they do. It's just made of chitin, so plants is cellulose and fungi is chitin. And then prototists, no, they don't. Although if you did tick that they did, then it doesn't matter for the marks in this one because I think there are some that are a bit ambiguous. For the carbohydrates, plants store it as starch, not glycogen, so that's a no. Animals, yes, they store it as glycogen. Fungi also store it as glycogen. Prototists, no, they don't. And then single-celled organisms, the only group here which is all single-celled is the prototists. Everything else, they can have single-cell organisms in their groups. There are single-celled plants, for example, there are single-celled fungi, there are single-celled animals like amoeba, but they are not all single-celled. Most of these are multicellular organisms. Viruses are another group of organisms. Explain why viruses are not classified with the other living organisms. So there's 2 marks here, so we need to give 2 reasons, 2 points, 2 reasons why the viruses aren't classified as living organisms with everything else. So kind of asking why are they not classed as alive. So to be a living organism, you must be able to go through and carry out all of the living processes or life processes. This might be something you've learned as Mrs. Gren to help remember it back when you were doing Key Stage 3. So living organisms have to do things like respire, grow, move, excrete, reproduce, and importantly reproduce like themselves, feed, so take in nutrients. They've got to do all of these things, and the main reason why viruses are not considered alive or don't get classified with everything else is that they do not do most of these things on this list. So we just need to be able to pick 2 of them and say they don't do these, and that's 2 reasons why we don't classify them with everything else. So I'm going to put that they do not respire. I'm going to put they do not reproduce, or they can't reproduce without a host. So that bit has to be on there important because they can replicate themselves technically, but they can't do it themselves. They have to get a host cell or a host organism to do it for them. Warfarin is a poison that has been used to kill rats for many years. Nowadays, there are many populations of rats that are resistant to warfarin. Use your knowledge of natural selection to explain the increase in the number of rats that are resistant to warfarin. So the rats clearly have evolved to be resistant, just like bacteria can evolve to be resistant to antibiotics. These are now resistant to the poison. So this makes sense when it's saying use your knowledge about natural selection. So we need to apply what we know about the process of natural selection, which we've probably learned in different contexts, maybe like about bacteria and antibiotics. But this time we're applying it and just using it to explain how rats could develop that are resistant. So how, going through the stages of natural selection, have that increased the number of rats that are resistant to the poison? So briefly, I'd write myself out a few bullet points just to remember the main steps of natural selection. So there's variation or mutation in the population, which makes some of them naturally resistant. These ones are going to survive and reproduce. They're going to be able to pass on that resistance genes or whatever it is, the genetics that they have that allows them to be resistant to their offspring. And then that repeats over many generations until we get large populations of rats that are resistant. Okay, so that's it. I've written 4 sentences for a 5-mark question, but I've got in every single thing that we need to say there to get the keywords and make the point. So we've got the word variation in there, which is the result of mutation. We've said that some rats will be resistant and they'll survive and reproduce. That phrase is really important. They pass on their resistance genes or DNA, or you've got other options for how to say that in the mark scheme, to their offspring, and that repeats over many generations. That's it. There's the main points here. The common mistake that people make when seeing these things like these 5 markers, especially when they are kind of describes or kind of explaining of a process, is that you've not just really thought about the fact that you might think you need to write loads and say a lot to get 5 marks, but a lot of biology is just about the keywords and making sure that you've got a process summarised nicely with the keywords, and the level of detail that you need is quite low as long as your keywords are correct and it's in the right order. So if any of these points weren't in the right order, and so that the process of natural selection could occur in that order, then you would potentially lose marks here. So that's something that's important to think about. But bullet points or short sentences with keywords used correctly is fine, and a small amount of that done really well will get you more marks than waffling for ages. The diagram below shows the structure of the human heart. Name the chamber labelled A in the diagram of the heart. So hopefully we know that the chambers are either going to be the atrium or the ventricles, and we're going to have to also say which side. So we need to know whether it's left or right. So always with heart questions, when you've got a diagram, put your left and right on first. So remember they're the other way round to your hands, and you can always remember which side is the left if you're looking at the picture, because it's going to have the thicker muscular wall at the bottom. Then always remember your atrium are at the top and the ventricles at the bottom. I remember it that A comes before V in the alphabet, so A goes on the top. But you can also think of the ventricles because they have this V shape. So they are V-shaped to the bottom, so that's why they are the ventricles. So A is therefore the right ventricle. So now it wants us to give the function of the structure labelled X. So this is asking us for the role or the job that that structure that's labelled plays between the atrium and the ventricles. And it is a valve. Doesn't really matter if you don't know the name of the valve, you just know it's a valve. And all valves carry out the same job, whether they're in the heart or they are in the blood vessels. They prevent the backflow of blood. So that's what we put as our answer for 1 mark. Coronary heart disease is a non-communicable disease that kills approximately 17 million people each year. Give an example of a risk factor for coronary heart disease and explain how it is linked to the disease. So a non-communicable disease is one of those diseases that doesn't spread from person to person, so it's something that normally is caused by lifestyle or genetics. And it's saying to give an example of a risk factor. That's something that increases your chance of getting that disease. And we want reasons to explain how the reasons that it is linked to the increased chance of that disease. So why does it increase your chances of getting coronary heart disease? So coronary heart disease is caused by blocked coronary arteries that are due to fat building up. This can be made worse if the blood vessels are damaged, or it's more likely to happen if the blood vessels are damaged. So the increase in fat in the blood that could cause the fatty buildup would be caused by high cholesterol, and the blood vessels could be damaged by high blood pressure. A risk factor for increasing the levels of cholesterol in the blood is that you could have a high fat diet. And there are multiple things that can increase blood pressure, but high salt in the diet, nicotine from smoking, or stress. So the high fat diet, high salt diet, nicotine, so smoking, and stress are all my risk factors. So I have to pick which one of those risk factors I'm going to talk about, link it to the high blood pressure or high cholesterol, and then link that to how that could increase the risk of coronary heart disease. So I'm going to choose high fat diet. So I'm going to say the high fat diet can increase blood cholesterol levels, and that increases the plaque buildup in the arteries, or the chance of plaque building up in the arteries. The other options are there in the mark scheme, and they're linked to the right explanation. So just make sure that you check that you've got the right risk factor and you've linked it to the right reason. The graph shows the trends in deaths from coronary heart disease. So we've got the deaths from coronary heart disease per 100,000 people up the side, and then we've got a line for men and a line for women. It wants us to compare the trends in heart disease deaths for men and women. So when we're comparing, we need to make sure we're looking at similarities and differences. And when it asks us to look at trends, we're looking at like differences in patterns over time. So we've got time on the bottom. So what pattern changes can we see? So they both increased from 2000 to 2003. And then after that, we get an overall decrease. So these are both similarities because they're doing them at the same time. The main difference is that the line for women is always lower than the line for men, which means the deaths from coronary heart disease is lower in women than in men. And so that's a difference. And then also look at the gap between the line. So this is something that's quite niche and I wouldn't necessarily expect everyone to have spotted this. But the difference between the number of deaths between men and women is actually getting smaller over time. So it was actually quite a big gap between the two lines, much higher in men. And over time it's getting smaller. We only need to give 2 points because it's only a 2-mark question. This could easily have been a 3 or 4-mark question though. But if we're only giving 2 points, we need to try and make sure that we're giving the similarities and differences. And we should always make sure we're including a data point as part of our description. So like the 2003 point here that we've read off the bottom of the graph, that's a nice one to include. So both increased in the number of deaths from 2000 until 2003, and then they decreased. So that's my similarities, and then my difference one is going to be that there are fewer deaths from CHD in women than in men. Explain how coronary heart disease can lead to the death of a person. So explain how you need reasons, like so how can coronary heart disease cause a death, but the process steps. So if it's a how, it's normally steps in a process, so linking things together. And you need to think about how coronary heart disease, what is actually going to cause the death of someone who has coronary heart disease. And normally that is a heart attack. A heart attack is the result of the heart muscle not being able to beat or contract. And that's due to lack of oxygen causing lack of respiration to provide the energy for the heart to beat. So we've got to then link this idea of coronary heart disease with this lack of oxygen. And so there's less blood being transported to the heart muscle because the coronary arteries are blocked. And so that means that's why we get lack of oxygen. So if you think about the order, we've got to talk about the fact that the coronary heart disease has caused the blockage in the coronary artery. So less blood is flowing to the heart muscle. That means less oxygen gets there. So there's less or no respiration to provide energy, and the heart muscle can't beat anymore, can't contract, and that's how we get a heart attack. So that's what we're going to literally put out in the right order to explain it in steps, making sure we use keywords like coronary artery, that the blood flow is blocked, lack of oxygen, lack of respiration, and the heart muscle can't beat. So if the coronary artery is blocked by fat buildup, then less blood can flow to the heart muscle. Without blood flow, there will be not enough oxygen transported to the heart muscle for respiration to take place. The heart will stop beating, causing a heart attack. So some common misconceptions and some mistakes that people make when they write answers about this, especially about coronary heart disease, is not saying heart muscle, just saying blood going back to the heart. Because blood obviously enters the heart in all the different vessels for the job and the function of the heart, you have to say specifically that it's the coronary arteries that are taking blood to the heart muscle itself, and that's the part that can't beat. Not saying that there's not enough oxygen, just saying no energy, or energy is being transported, they're all incorrect. It's about the oxygen not getting there in order to have enough respiration. So not linking that lack of oxygen to lack of respiration is also something that doesn't happen. As I said, saying arteries, not coronary arteries. If you don't say it's coronary arteries, then you won't get the mark. If you remember and think of it, they've said coronary heart disease at the top. So that is the part you need to make sure the keyword is in there. The student wanted to investigate how different concentrations of salt solution affected the mass of parsnip tissue. They set up the experiment as shown in the diagram below, with each parsnip cylinder in a different concentration of salt solution. So before we even read the rest of the question, parsnip tissue is just plant tissue. It's like a root vegetable like carrots, potatoes, and therefore I'm hoping that you would recognise if you're putting plant tissue into different concentrations of a solution, this is just the osmosis practical that you should be familiar with. Just because it uses parsnip and you may have used some other piece of plant tissue, potato or carrot or aubergine or pepper, doesn't matter what plant tissue you use, it's still the same practical. So part A says the student used a scalpel to cut the parsnip cylinders. Give one safety precaution that the student should have taken when using the scalpel. So when we do risk assessments for practicals, we should be used to doing a hazard. So what is it that could be dangerous? What's the risk to you that could happen? And then what's the precaution? This is the part where you write down what you would do to reduce the risk of harm or the risk of something happening. So in this case, we have to think about what would we do if we're using a scalpel. So the hazard is the scalpel. The risk is that it's sharp and therefore you could cut yourself. There is a risk of being cut. And so we need to give the precaution, what are we going to do or what should the student do when using the scalpel to make sure and try and protect themselves or prevent themselves from being cut. So hopefully you've done this a few times through all of your sort of years of doing science, and we should know that we should write down that we should cut away from ourselves, so you don't ever cut or point the scalpel towards yourself, or for example you don't put the parsnip in your hand and cut down onto your hand. You'd have to do it on a chopping board or onto the bench. So these are kind of the main two things I would expect someone to say in order to prevent themselves from getting cut when using the scalpel. We only need one of them here, so I'm going to say cut in a direction away from themselves. Some common mistakes that students make when answering these questions are saying things like use gloves. I would put a bet on, unless you're doing a dissection, you've never put gloves on in the lab at school. So that means that's not going to be an answer here. And it normally is never an accepted mark answer for things like equipment and practical safety. Just saying use it carefully or be careful with it is not enough. That's not telling you how to use it in a safe way. That's not descriptive enough. And saying stuff like don't put it near the edge of a table or don't walk with it pointing outwards and things like that. That's about generally being safe when sort of moving the scalpel around the lab, but not about actually using it. And it's asking about how do you use the piece of equipment safely. Okay, so part B, state 2 variables that need to be controlled in this investigation. So hopefully we know when it says controlled here, it's asking for variables that we would need to keep the same in the experiment. Control variables will always be things that could affect the results of the experiment if they're not kept the same. So it's always going to be things that could affect the processes or whatever's happening in your experiment. They're not going to be equipment-based answers. You're never going to get marks for saying use the same beaker, use the same tubes. That's not going to be what they want you to say. They want you to show that you understand that if you don't keep some of these things the same across your tubes or across the repeats of an experiment, then you wouldn't be able to compare your answers because the results would be affected. So there's some nice clues in the diagram here that we can use if we're not sure. So all of those cylinders are exactly the same size, or the same length as well in all of those tubes. That solution volume, the line is exactly the same in all of those tubes. So we need the same volume of solution, should have a similar or same size of length or some factor about the cylinders. It is a practical about osmosis. So things that can affect the rate of osmosis can also be control variables here because we don't want them to affect it. We are doing the concentration. So there's another factor that can affect osmosis, which is temperature. So you can have that as a control variable as well. Also, things that we can't really get from the diagram or thinking about osmosis is the fact that the time that they spend in the solution, the chips should all be in there for the same amount of time, because obviously if you left it for a longer time, more osmosis could happen. And also the same potato. So taking these cylinders from the same potato, or at least the same type of potato, the idea that then all of those cells have come from the same tissue so that they are the same, because they should have similar concentrations within the cytoplasm of their cells so that they should be as similar to each other as possible to make sure that they're having the same reaction. They would have all the same reaction in the different solutions. Common mistakes made when answering questions about control variables are saying the word amount instead of using a volume or time or some kind of measurement. So you need to say volume, you need to say size or length, not just amount of solution. Saying the independent variable. So this is something that people sometimes don't get right if they haven't read the question properly or they forget what control variable means. So we are changing the concentration of the solution in this practical on purpose. Okay, we've got different concentrations for a reason. That's our independent variable. That's what we're changing. So you can't keep that the same because that's literally the thing that we are changing. Everything else has to be kept the same, but that one thing is being changed on purpose. So that cannot be a control variable. So I'm going to pick 2 of these. I'm going to pick the volume of the salt solution should be kept the same. And you don't actually even have to say be kept the same because that is implied that you understand what control variable is. And also I'm going to put the time that the chip spent in the solution. You can obviously use some of the others that are all listed in the mark scheme. But just be careful. If you said volume, that's good, volume of solution. If you said amount of solution, then you don't get any marks. The mass of each parsnip cylinder was recorded before it was placed in the salt solution. When the chips were removed from the salt solution, the student blotted each cylinder before weighing it again. Explain why. So it's an explain question, so we need to give reasons why they blotted it. It's only 1 mark, but we have to make sure that what we're saying is going to get us that mark, because it's going to actually explain why they did that action. And it's going to have to come from a basis of like scientific knowledge. So not just what does blotting do, but you need to be able to justify why you are doing that step. So this is one of the examples where in practical questions like this, you have to be able to be justifying your method steps. So you need to be able to give reasons why you do various stages in the method and what the importance of that is to the actual experiment and the results. So blotting removes excess solution or liquid from the chip. Fine, but why are we doing it? That's not the reason why we're blotting. The reason why we're blotting it is so that we can make sure that any excess solution is removed, because it then doesn't affect the mass, because we are weighing them. So what we want to make sure is that we're only weighing the tissue and we're not weighing the tissue and some solution, because that then wouldn't be an accurate result. So there are different ways of writing this, and you've got some in the mark scheme. I've put to remove excess solution that could add to the mass. So if you just put to remove excess solution or excess liquid, that's not going to get you the marks. And equally, so that it doesn't affect the results on its own, won't get you the marks. You need to combine the two together to show that you understand that the liquid could be weighed and therefore added to the mass. Okay, so the student then recorded the masses in a results table shown below. And so we've got the solution concentration along the top, we've got the initial mass, the final mass, which they'd have done after blotting it, and then we've got percentage change in mass. So we need to calculate the percentage change in mass for the cylinders in 0.2 and 0.6 mole dm cubed. So the two gaps in the table, we have to work that out. And it did tell us that we're calculating percentage change in mass, so that's what we have to know how to do and use the formula. Okay, so to calculate percentage change in mass, we need to do the change, so the difference between the two values, and then divide that by the initial mass. And then as it's a percentage, we will have to times that by 100. So for the 0.2 mole dm cubed solution, we've got 12.2 was our final mass. We're going to take that away from the initial mass, which was 10.6, to get the difference, or the change in mass. So we increased in mass by 1.6. So then we're going to do 1.6 divided by the initial mass, which was 10.6, and then we're going to times that by 100. So that gives us 15.1%. And I have put it into the table, but as long as you've written it out and made it clear that this is for the 0.2 solution in your workings, then if you forget to put it in the table, it's not the end of the world. So for 0.6, we're going to do exactly the same thing. So we have a final mass of 7.9, take away the initial mass of 10.1. So we've decreased, we've lost 2.2 grams here. And so we're going to do 2.2 divided by the initial, which was 10.1, times 100. And that gives us negative 21.8%. So just try and make sure you keep those negative numbers in front of the minus 2.2, and then when you do it again, to remind yourself it has to have a negative in front of it, because it is a decrease, it is a loss in mass, and we need to make sure that we've got that correct. So if we get asked a subsequent question about it, we'll remember that that is a loss, like a minus, a decrease in mass. Okay, so I've brought the table over with our numbers in so we can use it for the next question. Explain the percentage change in the parsnip cylinder in the 0.8 moles per dm cubed solution. Okay, so 0.8 is the final column, and that is minus 29.8. So we need to give reasons why there was a decrease in mass at the 0.8, or the chip of the 0.8 solution. So it didn't say decrease in the question, it just said percentage change. So we are obviously using this negative number, and we've got the table information to know that it was a decrease. So why did it lose mass? What are the reasons? So there's 3 marks on offer here, which seems quite a lot, but that's because this explanation can take a few stages. So give reasons why the chip lost mass. Why did it lose mass? Well, because it lost water. Why did it lose water? Well, because the water moved out of the cells in the chip by osmosis. It's really, really important, because at no point in this question have they mentioned the word osmosis. You have to know this is about osmosis, and this is the osmosis practical, and you should be using this key word in your answers. So that's not enough to get us the 3 marks, because why did the water leave the cells by osmosis? Using our knowledge of osmosis, we have to be able to explain why that happened, and why it happened specifically in that solution concentration. So the reason why water left the cells by osmosis is because there is a higher salt concentration outside of the cell than inside of the cell. Okay, so we know that water moves from a dilute solution to a more concentrated solution. That is our definition of osmosis. So that is the reason, that explains why the water left the chip cells and went out into the solution, because it has a higher concentration than the cells inside the parsnip. So my answer is going to be that the chip in the 0.8 solution decreased in mass because water moved out of the chip by osmosis. This is because the solution outside of the cells was more concentrated than inside of the cells. So I've got my water moving out, I've got the process and saying that it is osmosis, and then I've got my reason because of the difference in concentration. So common mistakes are not saying the word osmosis, so not mentioning the process, not saying that key word and losing out mark. Not saying the reason why the water moved out, so not explaining that difference in concentration, why osmosis has happened. And not saying where the water's moving to. I always tell my students, like, so if you're doing an osmosis question, you just say it's osmosis for start. Also tell me, where is the water going? Okay, so if osmosis is happening, water must be moving. So where is it moving? In this case, it's moving out of the cells because we've got a decrease in mass. If it was an increase in mass, you'd be talking about the water moving into cells by osmosis and not missing out that key thing, and then explaining why it's going in that direction. The student repeated the experiment but used a crinkle cutter to cut the chips instead. A crinkle cut chip can be seen in the image below. Explain why these chips showed greater changes in mass than the cylinders. So we need to explain why, so we need to give reasons why there were bigger changes, so more increase or more decrease in mass. And you need to compare them. So we need to compare these crinkled cut chips to the cylinders. What's different? So I've drawn a cylinder next to it so you can see this idea, but there has to be something different about the crinkled cut chip compared to the cylinder that could affect osmosis, because more or less osmosis is happening because it's told us there's greater changes, more decreases or more increases in mass. So that means more water is moving in or out of the chip cells. So that means that something about the crinkled cut chip has affected the rate of osmosis. So the chips, the crinkle cut chips, have a bumpy or uneven surface. So they're kind of all folded and bumpy. They've got these kind of ridges as opposed to being smooth and straight like the cylinders. So if we think about the three things that can affect the rate of osmosis, we've got temperature, concentration gradient, and surface area. So clearly, this image has been given to you for a reason. The description of it being crinkle cut there is a reason, and this is about surface area. We're not changing the temperature, we're not changing the concentration gradient. All they changed was the surface area of the chips. So if they've got more surface area, then osmosis is going to be happening faster, or more water can be moving across the membrane in the same amount of time, and that explains why there is greater decreases or increases in mass, because more water is moving, because there's an increased surface area in the chips compared to the cylinders. So that's our sort of point for 2 marks. We've got to say what's different, and it's the surface area, and then say how that has caused more water to move. So for our first mark, we're going to explain that the new chips had a greater or larger surface area. And then we give the reason why that affected the greater changes in mass. So more water moved out and into the chips in the same amount of time. The more water part is important there, or talk about the faster movement of water in the same amount of time. That can also get you a mark here as well. The passage describes the cycling of carbon in ecosystems. Complete the passage by writing a suitable word in each of the spaces. So the cycling of carbon in ecosystems is just a long way of saying it's the carbon cycle. So when you get these kind of fill in the gap long paragraph questions, and they've not obviously given you the words, so you have to think about them yourself. So I would just jot down some kind of key words that I would think of when I think about the carbon cycle. And then you've got that list there then that you can kind of refer to, because it's going to be keywords and it's going to be words that we know. So the carbon cycle involves death and decay, which is done by decomposers. It involves respiration. It involves photosynthesis. These are all kind of the main processes in the carbon cycle. And then we've got like the combustion or burning of fossil fuels and the formation of fossil fuels. So all of these are kind of like the key things I would think of and the key words I would think of when I think carbon cycle. And this could be the same depending what the topic is, the kind of long answer is about. So nutrients return to the soil when organisms die and tissues do something. Tissues decay, right? Nutrients return to the soil. So the breakdown of dead matter returns that nutrients back to the soil. That's the process of decay. Dead organisms are broken down by something, usually in warm, moist conditions. So it needs to be an organism that breaks down dead matter, right? So my initial thought would be to put decomposers in there because it's breaking down dead organisms, and that's what decomposers do. Sometimes you could put these in kind of pencil underneath the line before you make a decision and then come back to it if you change your mind. This is something I think would be a good idea. Organisms which feed on dead matter are known as what? So, we can't use decomposers twice because we need a different suitable word. Decomposers are the organisms which feed on dead matter. So, decomposers needs to go into this gap. So we need to think of something else that we can use. So dead organisms are broken down by what? And they've put warm, moist conditions. So these are conditions that allow certain types of organisms to thrive and grow. And so we need to mention like a type of organism that is a decomposer. So bacteria or fungi. That's why that word goes in there, because it's talking about the conditions that they grow in really well. The decomposers use something to break down the tissues which release nutrients into the soil. So they're using something that's in the decomposers that break down tissues or break down substances and release nutrients. So they actually obviously digest that material because they're feeding on the dead matter. So in this case, what actually breaks down biological material? It's the enzymes. So it's the digestive enzymes that the decomposers release in order to break it down. And then it releases nutrients into the soil and something into the air. So what do decomposers release into the atmosphere? And if you think about it, this is all about the carbon cycle, and so far we haven't mentioned carbon yet, but hopefully we know that decomposers, when they break down, when they decay material, they obviously respire and they release carbon dioxide through the process of respiration. So it's got to be carbon dioxide being released into the atmosphere. And that makes sense because of the sentence, but also because this is about the carbon cycle. So I've crossed off some of the things we've used already. So death, decay, and decomposers have all gone off of our keyword list. So processes that return carbon back into the atmosphere include something and combustion. So we need a process that releases carbon dioxide that's not combustion, so not burning of wood or something or fossil fuels. So the only one on our list that releases carbon dioxide into the atmosphere is respiration. The only process that removes carbon dioxide from the atmosphere is, hopefully this is something we just know, it is photosynthesis. So photosynthesis is the only process that actually takes carbon dioxide out of the air because plants absorb carbon dioxide. The cycling of carbon is in balance. Humans have been cutting down trees and burning something which has disrupted the carbon cycle. So what have we been burning? So we've talked about combustion on our list, but the word combustion has already been included further up. So we need to be burning something which has disrupted the carbon cycle. So it's the burning of fossil fuels. That's obviously releasing more carbon dioxide. Humans have processes that help to regulate their body temperature. Name the process that maintains stable internal conditions in the body. So this is effectively a backwards definition question. Instead of asking you to define something, they've basically given you the definition and asked for the word. So the process that maintains stable internal conditions is homeostasis. Explain why humans need to maintain their body temperature at an optimum level. So this is an explain question, so we're giving reasons why the temperature needs to stay at an optimum level, or at 37 degrees. So this is the question where we can sort of think about what would happen if it was too low or too high. What's the main reason for homeostasis mechanisms controlling temperature? It's to do with enzymes. Okay, so enzymes need an optimum temperature to work. And the classic is that if it was too high, the temperature increases too much, then we can denature the enzymes and they will no longer work. They're needed for chemical reactions to happen at an optimum rate. So you could sort of add an extra why is enzymes important. But we only need 2 marks worth here. So saying that enzymes need to be at the optimum temperature to work best, and that if it got too high, then they could be denatured is enough for 2 marks. The diagram shows changes in an artery that happen as part of a temperature regulating process. So we've gone from having 0.86 millimeters as the lumen to having 0.18 millimeters as the lumen. So the lumen is getting narrower or smaller. So as part of the temperature regulating processes, which bits of temperature regulation do we know involve blood vessels? Because that's what this question is going to be about. So when we're talking about temperature control in the body, we talk about vasoconstriction or vasodilation, which is the changing in the width of the lumen of blood vessels. We know from this picture that it's about getting narrower, narrowing the lumen in the blood vessel, so therefore it must be about vasoconstriction. And the first question asks us to give the term used to describe this change in artery shown by the diagram, so it's just vasoconstriction. Calculate how many times smaller the area of the lumen is after the change has occurred. So in order to compare the area differences, we need to calculate the area for both. And then when you're doing a how many times smaller question, you divide the larger one by the smaller one in order to find out how many times it would fit into that number. And then that tells you how many times smaller it is. Because we need to calculate the area of a circle, we should know that to do that we need pi r squared. This is a maths skill question, and it's told us to use 3.14 instead of pi. So to use 3.14 in your calculator instead of using the pi button. And it wants us to give our answer to the nearest whole number. This is important. We may not get all of the marks if we don't do that as part of our answer. So it's always worth making sure that we check back at the end that we've done everything it's asked. So I'm going to write down pi r squared because I know I'm going to use that formula. And also means that the examiner also knows that I know how to calculate the area of a circle just in case something happens. So I'm going to use 3.14 just for pi, like it said in the question. So now we've obviously got the diameter in the question. So we need the radius for pi r squared. So we're going to do the diameters in the question divided by 2. And then we're going to square that. So we're going to work out the radius. You can do it by doing 0.86 divided by 2 first and then putting it into the formula. Same for 0.18, you can just make it 0.09 and then put it into the formula. But that's an extra step in case you go wrong. So you can just do this and put it over 2. So for the first one, we're going to do 3.14 times 0.86 divided by 2 squared. And that gives us 0.581. And then for the second one, it gives us the area as 0.025. Now, I've not put units on these, but obviously it would be millimeters squared. And then to work out how many times, like I said, we take the larger number, divide that by the smaller number to work out how many times 0.025 would fit into 0.581. So that gives us 23.24. But remember, it's asked us for the nearest whole number. So because it's less than 5 after the decimal point, we round down and we just stick to 23 as our nearest whole number. If you rounded the 0.581 and the 0.025 before you did your division, then you may end up with 19 times. So either of those two answers is correct, because if you rounded those two numbers correctly before you did the division, then you can get 19. Anything between 19 and 23 is not correct, because if you rounded incorrectly, then you will not get the marks. So you would just get 2 marks if you've done the calculations of the area correctly, but then if you rounded incorrectly and then got the wrong answer when dividing, then you don't get the marks. So just be careful with rounding. I would say don't do it until the very end, just to be on the safe side. Explain how this process helps to increase body temperature. So the process it's referring to is obviously vasoconstriction. And how does it help to increase body temperature? So hopefully we know that it's because it reduces heat loss. So we need to explain how this process, so explain what actually happens and then explain why that helps to reduce heat loss or helps to increase body temperature by reducing heat loss. So first of all, we need to give the explanation of what happens. So how does it actually happen? So the walls of the arterioles contract to narrow the lumen. So actually describing what's happening in vasoconstriction is going to get us a mark here because it's not given us any of that information other than just showing us the diagram. So we are explaining that we understand what vasoconstriction means. What does that then do? That reduces blood flow through the blood vessel. So that's the how. Now we need the explain part. The why does that help to increase the body temperature? Because it reduces the amount of heat energy lost through radiation from the blood. And that's because these blood vessels, where it's happening, are close to the skin. The constriction of the blood vessel walls narrows the lumen. This means less blood flows through these vessels near the skin. this reduces heat energy lost through radiation. Okay, so you've got 3 bullet points, and there's obviously variations in these, the language you can use. Even if you said something slightly different, that should be fine. And we've done our 3 things for 3 marks, and sort of we've explained the process and then how or why it helps to reduce heat loss. The diagram shows a neuron. Name 3 structures that are found in plant cells that would not be found in this cell. So hopefully we should know that a neuron is an animal cell, but you can see that by some clues in the picture as well. But really, this question is then just asking you what are the 3 unique structures to plant cells that we can't find in animal cells. So it's the cell wall, chloroplasts, and a permanent vacuole. Name the type of neuron that sends signals from receptors in the eye to the brain. So when you think about the types of neurons, there are 3. There's sensory, motor, and relay neurons. So it's asking us which one of these 3 it is. And it has to be the type that will attach to receptors. So sensory neurons are attached to sense organs. That's how I remember it, sensory, sense. So because they're attached to sense organs, they're attached to receptors that we find in sense organs. And obviously the eye is a sense organ. So it must be sensory neuron. The diagram shows light rays entering a person's eye from a distant object, so far away. Explain how the eye can adjust to form a clear image of a nearer object. So we need the process steps and why. So how would it actually change and why would it change to accommodate a near object instead? And it says adjust, so we need to talk about the changes in the structure of the eye that would make this happen. And obviously it's happening for a nearer object, so the light would be coming from a closer or an object closer to the eye. So when we do that, instead of having these straight lines, we would get kind of more diverging light from a close object coming towards the eye. So in order to then refract and bend those light rays back towards a point on the retina, we would need to refract those light rays more. So we need a thicker lens. So it's about how the eye is going to make the lens thicker and then explaining that that would refract the light more in order to focus it on the retina. So our first mark is about the ciliary muscles contracting, and then we need to talk about the suspensory ligaments loosening. These are the 2 ways round that we need to get it right, and we need to just remember if we're doing a near object, ciliary muscles contract, suspensory ligaments loosen. I like to think of it about C for ciliary muscles contract, and then ligaments L loosen. This would make the lens thicker, so the light is refracted more to converge on the retina. So there's lots of keywords to include and get the right way around here. So the ciliary muscles contracting, the suspensory ligaments loosening, talking about the lens getting thicker, refracting the light and it converging on the retina. And this is kind of a little paragraph we need to learn for near objects and far objects. So we just have to learn it, make sure we get it the right way around. And it's kind of just a sort of a repetition of facts that you have learned in order to explain these 2 processes of like accommodating for near objects or accommodating for far objects. Variation can influence the phenotype of an organism. Put a tick for each row to show what type of variation causes the following phenotype differences. So we've got to decide if each of these is caused by just genetics, by just the environment, or by both genes and the environment. So if we go through these one by one, blood type is definitely caused by genes. You inherit that from your parents. Height is both genes and the environment because it can be affected by your diet as well as genes. Genes coding for flower colour because again that's kind of like eye colour and hair colour in humans. And then the language you speak is definitely just due to the environment that you're raised in, where in the world you are raised. That is determined if you speak a certain language. It's nothing to do with genes. Genetic variation in a population is due to mutation. Define mutation. So this is just a definition. We have to know just how to define a mutation. So it's a change to the DNA. You've got some alternative wording in the mark scheme you can use as well. Some plants, like garlic, produce antimicrobial substances within their tissues to help protect them from insects. Scientists are investigating ways to genetically modify barley plants to produce similar antimicrobial substances. State the term used to describe organisms that have been genetically modified. So organisms that have been genetically modified have had a gene from another organism transferred into their DNA. Literally think about gene transfer, so the term we use is transgenic. Explain how scientists could use a gene from garlic to genetically engineer other crop plants to produce the same antimicrobial substances. So explain how it wants the steps in the process, so how would they actually make this happen? And if we're genetically modifying something or genetically engineering something, we're cutting a gene, and in this case from the garlic plant, and we're taking that gene out and we're going to transfer and put it into a plant that doesn't normally have that gene. So this is testing us on our ability to remember the stages of genetic engineering. So we're going to need to cut the gene out, and we're going to need to mention restriction enzymes in order to do that properly. This will create sticky ends. We should mention these. Using the plasmid as a vector is another thing we should mention, and then the ligase enzyme that we need to join the DNA into the plasmid. And then we've got to obviously transfer it into the plant, but we would need to do that at an early stage of development if we wanted the whole barley plant to be able to make these substances and not just a few cells. Scientists would need to cut the gene that codes for the antimicrobial substance out of the garlic DNA using restriction enzymes. A plasmid vector would also be cut using the same restriction enzymes so that they had complementary sticky ends. The garlic gene and the plasmid DNA can then be joined using the enzyme ligase. The garlic gene can then be transferred into the barley cells during an early stage of development to ensure the plant can make the substances all the way through. So we need to make sure, obviously, we're putting in all of those key points, which I've done here to get the 4 marks. So although I've written them in full sentences, you can see from the mark scheme that the kind of really key points you need are cutting the gene out using restriction enzymes, talking about a plasmid vector, and talking about using the same restriction enzymes to get sticky ends so that they match, and then joining the DNA and the plasmid vector using the enzyme ligase, and then ideally that would get us 4 marks. We also obviously need to mention the early development when you're transferring it. So if you're doing a genetic engineering question, you have to talk about the transfer, where's it going from and to, and if it's plants, we need to make sure we're doing it at an early stage of the development. Explain why farmers want to produce the genetically modified crop plants. So we need reasons why. There are 3 marks here, but we might not need 3 reasons, and we'll talk through why in a minute. And why would they want the genetically modified plants? So remember, the genetic modification we're doing here is that they would have some resistance to pathogens because they'd be able to produce this antimicrobial substance. If we think about farmers and what they care about and why they want something to change specifically about a plant, they would always want a greater yield. So that's the mass or mass of plants that they are going to get that they could sell or use to eat themselves. It depends on whether they're a commercial farmer or not. So if we think about the fact that we're creating genetically modified plants that have resistance to some pathogens, how can we link that to yield? Well, less damage is going to happen to the plant if they are not going to be getting infected with pathogens, and therefore the farmer's going to have a greater yield. So there are 3 points. So explaining that by having these genetically modified plants, they're going to be resistant to pathogens. Therefore, there'll be less damage to the plants from pathogen infection, and therefore the farmers would have a greater yield, which is something that they want because it means they can get more profit, or they've not wasted that part of the land that they've been using to grow that crop. So our answer then can literally be 1 sentence, but it can get us the 3 marks. So because plants that are resistant to pathogens would be less likely to be damaged, so the crop yield can increase. That's it. A student wanted to investigate whether there was more carbon dioxide in inhaled air or exhaled air. They set up a basic respirometer with one-way airflow like the diagram below. When the mouth breathes in, air from the atmosphere is pulled in through tube X and moves through flask A. When the mouth breathes out, the air flows out through flask C and then out through tube Y. Explain what you would expect the lime water to look like in the flasks A and C after 30 minutes. So this practical is all about looking at the carbon dioxide concentration in the air we breathe in and the air we breathe out. So the question wants reasons why. They want us to explain why the colour might change in the lime water. So we should hopefully know that lime water, what lime water is, and it does again from chemistry, but also this is a common substrate used in respiration practicals like this. And it's 4 marks. So we need to give 2 marks for what we'd expect to see in the flask, each one, and then 2 marks for the reason why we'd expect to see a change or not in each flask. So hopefully we know that lime water goes from clear to cloudy with carbon dioxide dissolved in it. And hopefully we know that there should be more or a greater concentration of carbon dioxide in the air we breathe out than the air we breathe in. And this is obviously due to respiration. So this is what we'd expect to see. A should stay clear because there's very little carbon dioxide in the atmosphere that's just being drawn through that tube. But C, which is after the mouse and therefore contains the air the mouse has breathed out, should go cloudy because there's more carbon dioxide in the exhaled air coming from the mouse because the mouse has been respiring and it needs to get rid of that carbon dioxide as it breathes out. So we have everything now to write our answer. So flask A would remain colourless, but flask C would turn cloudy. That's 2 marks, 1 for saying what we'd expect to see in each flask. And then our explanation can just be 1 sentence and it should still get us the 2 marks because we just need to say, because there's more carbon dioxide in the air exhaled by the mouse than there is in the atmosphere. Suggest how this experiment could be changed so that the rate of respiration could be calculated. Include any extra apparatus you would need to use. So this is a suggest question. This is your own idea or your own knowledge. It's not just a recall factual question. So you need to use your own ideas and knowledge that you can kind of get from the clues in the question, but also what you've learned. And they're asking what needs to be changed. So what needs to be different? What do we need to do with this setup? Because it's asking about apparatus. That means it's asking about pieces of equipment that might need to be different in order to calculate the rate of respiration. So remember, rate equals change over time. So we would need to measure something, get a number, measurement, and then divide it by the amount of time. So if we're doing respiration, we want to measure the rate of respiration. Normally we do that by collecting the products. So collecting the carbon dioxide, instead of just letting it go into flask C, collecting the volume of that in a set amount of time. So what piece of apparatus could you use to collect the gas and measure the volume of it? And then what else would we need to use to measure time? These are the bits of equipment it's kind of leading you towards trying to get you to say, because it should be sort of knowledge about respiration practicals, how to calculate rate, what you need in order to calculate rate, and then what pieces of equipment you can use to do that. So my first thought would be to attach a gas syringe to that tube that's leading out of flask B. So we don't need flask C anymore, and we would attach a gas syringe there to collect gas coming out of flask B. And any carbon dioxide from the air coming in would have been got rid of through flask A. So we're measuring the volume of air being breathed out. We need to measure the time as well. So we need some kind of timer or stopwatch. So my first mark is going to be from saying I would collect the carbon dioxide gas exhaled using a gas syringe. You could also do a measuring cylinder like underwater with you bubble it up. That can also work as well. And you would need to do it in a set amount of time, as we said, so say a timer or a stopwatch. The students repeated the experiment without the mouse. Describe what you would expect to see in flask A and C after 10 minutes. So we're not giving any reasons here. It's not an explain question. It's just a describe question. So it's asking us what would happen when there's no mouse in there. So there's nothing respiring. There's no carbon dioxide being produced. So what would we expect to see in the flasks? So hopefully we can obviously see it's quite an easy 1 mark question here. We just need to say that the lime water in both flasks would stay clear or does not go cloudy, because we would expect nothing to happen because there's no carbon dioxide coming from a source anymore. There are 2 types of respiration, aerobic and anaerobic. Complete the word equations for the 2 types of anaerobic respiration. So immediately it's helpful that it's in bold, but immediately I'm thinking no oxygen when I see the word anaerobic. That's what that means. So I'm going to note that down to remind myself. It's the word equation, so no symbols, no cheating by just writing symbol equations instead. So the first one says in animal cells, glucose breaks down to what? It is lactic acid. And in plant cells, it's glucose breaks down to ethanol and carbon dioxide. So this is, again, it's something we should just know. We should know the difference between anaerobic respiration in these 2 types of cells. We should know the equations. This is just factual recall. You can put alcohol instead of ethanol. Sometimes that is allowed, but it should be ethanol. So now we've got some questions about those equations. Anaerobic respiration can only sustain exercise for a short time. Explain why. So if we talk about exercise, this is where we're talking about it in animal cells or human cells or muscle cells. So we need reasons why we can't exercise for a very long time if we're only doing anaerobic respiration in cells. And what we mean by exercise is muscle contraction. So realistically, why can't muscles contract for very long if we're doing anaerobic respiration? So hopefully we know from our bit of content knowledge about anaerobic respiration is that not a lot of energy is produced through anaerobic respiration compared to aerobic respiration. And obviously muscles need energy for muscle contraction. So we can only do it for a short amount of time before the muscles will stop contracting or become too weak or too tired because not enough energy is being produced from anaerobic respiration. So muscle contraction stops because not enough energy is being released. Anaerobic respiration in yeast cells is important for the food industry. Explain why yeast cells are important to the brewing and baking industries. Okay, so it's an explain why, so we need to give reasons for each one. So reasons why yeast cells are important for brewing and reasons why they're important for baking. So in brewing, that means it's used to make beer and alcoholic drinks like wine, and in baking, it's used to make bread. So that's what it's used for. We need to explain why it's used for those industries. So why do we use yeast? So this is about the other equation. So this is the reason why they get you to do the equations first. It gives you kind of a bit of a clue. Because anaerobic respiration in yeast is very similar to anaerobic respiration in plant cells. So it's going to produce ethanol, which is an alcohol, and that's what makes the alcoholic drinks alcoholic. And the carbon dioxide is useful because it's going to make the bread rise. So those are our two reasons. So we know what they're used for in those industries, and then we're giving a reason why they're used. The anaerobic respiration of yeast produces carbon dioxide, which makes bread rise, and it produces ethanol, which is used to make alcoholic drinks. There we go. That's it. It's actually just one sentence, but it will get us 4 marks. And this is really just kind of content recall that we should have learned when we learned about fermentation. Beta thalassemia is a recessive inherited disorder which reduces the production of hemoglobin in the body. The family pedigree shows the inheritance of beta thalassemia in one family. So my first thought is, what is a recessive inherited disorder? It's the one that needs 2 alleles to be the same to be present in the phenotype. So it's the one where you have 2 lowercase letters, for example, in order to have the disease. And if we're thinking about maybe a question we might get asked later, we might not, but I'm thinking, okay, if it reduces the production of hemoglobin, that means there'd be less oxygen being transported if we don't have as much hemoglobin in the body. So we've got the parents in the first generation, then we've got their children and their grandchildren in the third generation. So it wants us to state the sex chromosomes of person B. So if we have a look, obviously sex chromosomes are XX or XY, which is the options we've been taught, and we are using the key here to see that B is a female with beta thalassemia because she's represented by a grey circle. So therefore, in terms of the sex chromosomes, the only thing we care about really here is that she's female, so she must have XX. Explain why sperm determine the sex of offspring at fertilization. So the reason why is what we need to try and say here. Why does sperm chromosome determine if an embryo has XX or XY? And this is simply because sperm can either contain an X or a Y, depending on which chromosome is present out of the 2 options. Eggs can only contain X chromosomes because female cells can only donate an X chromosome to each gamete. So if you have a Y chromosome present in the sperm, then the fertilization will happen and you'll get XY and therefore a male offspring. If you have an X in the sperm, then you'll get XX and you'll have a female offspring. So this is basically what we need to say. We need to say that sperm can carry a Y or an X, and having a Y chromosome determines male, and if you don't, then you're female. Explain why both individuals F and G in generation 2 must be heterozygous for beta thalassemia. So what are the reasons why both F and G must be big B, little b? So if you find F and G, we can see that they do not have the disease, but they have had an affected child. So this is very common with recessive disorders. In questions like this, they'll get you to go and find two parents that do not have the disease and they've had a child that does have the disease. So that must mean they both have to be heterozygous because they both must have a copy of the lowercase allele, the recessive allele, in order to give both of them to give a copy to child I in order for child I to be little b, or double or homozygous recessive. So we can always start our explanation by saying they are unaffected, but they produced a child, in this case child I, who is affected. So we can say they must both have had at least one recessive allele in order to both pass on the recessive allele to child I, but obviously they are both unaffected, so they must also have had a dominant copy. Use the Punnett square to calculate the probability that the next child, individuals F and G, could have beta thalassemia. So we know from the earlier part of the question that both parents are heterozygous, so we can really easily put their gamete alleles into the Punnett square. I've put F and G on there just so that we know, although it's the same, so it doesn't really matter. And then we fill it in as normal, so 2 capital B's, 2 squares that have large b and lowercase b, and then 1 square with 2 lowercase b's. So that then needs to be circled to show that we understand that that genotype would produce an affected child, and then we need to give the probability. So it's 1 out of 4 or 25% chance that the next child they have will have the homozygous recessive and therefore have the disease. So as you can see in the mark scheme, just doing the Punnett square without circling the little b and giving the information doesn't get you the mark. So make sure you add this every single time you do a Punnett square question. You need to make sure you circle the one thing that the question was asking you to look for the probability for and then label it with the probability. Explain why it's important for people like C or E to have a pedigree analysis in case they decide to have children in the future. So we can see C and E are children of A and B, so their mum has beta thalassemia and so does their brother. It says use percentages or ratios to help illustrate this. So we're going to need to sort of explain what the percentage chance might be of them passing it on depending on what the analysis shows. So why is the reason why they like to have a pedigree analysis? And it's because obviously they might be able to pass on the recessive allele. So they're going to want to know what the probability is of their offspring having the beta thalassemia allele. So if we think about the percentage chance, obviously we've just seen that it could be 25% chance of having a child with beta thalassemia. There could be 50% chance of passing on a recessive allele if you are heterozygous. And if they're homozygous dominant, so they have 2 capital B alleles, 2 dominant alleles, then there's 0% chance of them passing on the affected allele to their offspring. So it's all going to depend on what alleles they have and then what alleles their partner has in order to determine what the probability is of having a child with the beta thalassemia allele or having the disease, but only with analysis will they be able to show that, so then they can obviously make those decisions. So analysis will determine the probability of passing on the recessive beta thalassemia allele. It could be 0% if they are homozygous dominant, or up to 50% if they are heterozygous. A father with the genotype capital B, so homozygous dominant, and a mother with the genotype little b, or homozygous recessive, plan to have several children. Explain why none of their children will have beta thalassemia. So explain why none of their children, the reason why none of their children will be little b. And when it comes to these questions, it's often just easier to draw a Punnett square rather than trying to write it out in words. And often you will get marks for that as long as you annotate it correctly. So let's put those into a quick Punnett square. So we've got 2 capital B's and 2 little b's. So it shows that 100% of their offspring are going to be heterozygous. So they're going to all have at least one dominant allele and one recessive allele. So they'll all be carriers, but none of them will be homozygous recessive. So none of them will have the disease. So you can say none of them will have 2 recessive alleles. You could say they can only get 1 recessive allele from mum, and so therefore they can't therefore have 2 because the father can only donate capital or dominant alleles. However you want to explain it, you've got some options in the mark scheme, but if you want to do the Punnett square, that's fine, but you would need to have this extra little bit of text that I've written next to it just to make it clear that you understand that 100% of the offspring are heterozygous, or something like no child will have 2 recessive alleles to have the disease. A spirometer is a piece of equipment that can be used to measure the volume of the lungs by measuring the volume of air breathed out after a single large breath in. So the patient takes a large deep breath in, they breathe out as much air as they can into the spirometer, and the piston moves up and pushes the needle along the scale. The reading on the spirometer can be compared to normal reference values to see if the patient's lungs are functioning normally or not compared to normal predicted values. This would be for something like the predicted values for their age, height, mass, etc. Athletes who do vigorous exercise daily as part of training have high physical and aerobic fitness. Measuring a person's lung volume can indicate their fitness level. Design an investigation using the spirometer to find out if daily exercise increases a person's lung volume more than weekly exercise. Include experimental details in your answer and write in full sentences. So hopefully we'll be familiar with the idea that any design and investigation question is a CORMS question. And we're clearly comparing the daily exercise versus a weekly exercise. So CORMS, the C stands for change, which is the independent variable, so what you're going to change in the experiment. The O stands for organism, so what biological organism are you going to be using. R stands for reliability. M stands for measure, or the dependent variable, what will you measure in the experiment. And then S stands for standardization, which is basically the control variable, so what we're going to keep the same. So it's telling us we need to use the spirometer, and it gave us a really important breakdown of how the spirometer worked and that we can measure lung volume because we can measure the volume of air breathed out. So that's going to be our dependent variable. It tells us we want to compare the daily versus the weekly exercise. So we're changing the amount of exercise they're doing. Is it every day or just once a week? We're obviously going to be using people, humans, for this experiment, but we need to think about what we should keep the same or similar about those people. So similar age bracket, BMI, fitness level, or health defects and things like that. We should mention when and where we're going to do repeats of the experiment. In terms of thinking about standardizing, how long they do the exercise for every day or once a week, the type of exercise they do, so it needs to be the same, whether it's star jumps or running for a certain distance or whatever it is. And how long are they going to do it for? So are they going to do it for a month? Are they going to do it for 2 months? When are you going to measure them before and after the exercise? How long is the time gap? Okay, so my first sentence is for a large group of people with similar age and BMI. So that's my people, that's my organism, and what's similar about them as well. So that's a bit of S. We should measure their lung volumes using the respirometer. Okay, so I've said I'm going to use it and what I'm going to measure. I improved that a little bit by actually saying we're going to measure the volume of air they breathe out in centimetres cubed using the respirometer. So I've been more specific about what I'm going to measure and the volume, the units. They should do this 3 times and an average should be taken. So that's my reliability. So I'm not just taking one lung volume measurement, I'm getting them to do it 3 times and then I'm going to average it. Then the group should be given a weekly exercise to complete. So once a week they do the exercise. All participants should do the same exercise for the same amount of time each week. So there we go. I've said what I'm going to keep the same in about the exercise. After 2 months, their lung volume should be measured again. So now I need to talk about the comparison. So a second group with similar age range and BMI would do the same, but have to do the same exercise for every day instead of every week. So then I've got the comparison between the daily versus the weekly exercise. So that's what I'm changing. And then the average difference between the volume of air breathed out between the before and after exercising for 2 months can then be compared between the two groups. So I'm obviously going to have all of the people in the large group, their average before, and then all of the people in that group, their average after doing the daily exercise versus the same numbers for the group that did the weekly exercise. And then we can see which one improved the most. So you can see from the mark scheme, there's lots of things that you can say. There's lots of other things and variations that you can mention. So your answer is going to look potentially very different to this, but it has to include all of those things. So you have to have talked about all of the C, O, R, M's and S. You have to have at least one of each of those to make sure that this account would be sufficient to get the full marks. Tuberculosis, or TB, is a type of lung disease caused by a bacterial infection. The infection destroys lung tissue. Name what type of drugs patients would be given by doctors to treat tuberculosis. So if we didn't already know, TB is caused by bacteria, so hopefully we should know what type of drugs kill bacteria. That is antibiotics. Infection with TB can influence lung volume. Spirometer measurements taken from patients who had multiple infections with TB can be seen in the graph below. So the white bars are before they had TB, the grey bars, the light grey bars are after the first TB infection, and then the dark grey bars are after their second TB infection. And they've measured the mean volume of air breathed out in litres, and that's clearly taken from the spirometer. And then we've got different age groups that have been tested. So it wants us to describe the trends shown in the graph. So trends just means patterns. So what patterns can you see in the data? And it's just a describe question, so we're not trying to explain the differences we can see, just saying what we can see in terms of the differences in the patterns. So the main one is that there's consistency across all the age groups where between before TB and after the second infection, the more infections you have, clearly, it decreases the lung volume. We've also got a general overall decrease in lung volume as you get older. Also, even if just you have one infection with TB, it's reducing your lung volume. So we've got these kind of three things. So having TB reduces your lung volume. The more TB infections you have, the lower your lung volume, and overall your lung volume decreases with age. And these are the 3 points that can give us the 3 marks. Always look for overall trends in graphs like this. So this is an big slope you can see down between these bars, but then also thinking about what's happening within individual groups and between comparing individual bars is also the best way of spotting all these patterns and getting enough detail. So infection with TB reduces the volume of air breathed out, and the more infections you have, the lower the volume. Lung volume also decreases as age increases. This is just a way of writing those 3 points that we mentioned, but in a way where we're talking about patterns. So as this changes in this way, is a good way of kind of writing out patterns in data that you see from graphs. The infection damages the alveoli. Explain why people who have had tuberculosis may find exercise difficult. So infection with TB is obviously being referred to because it's the next part of the question. It damages the alveoli, which is the site of gas exchange in the lungs. Why might we need to link, or how could we link gas exchange and exercise? Well, when you're exercising, you need more oxygen for more respiration because you need to release more energy for your muscles to contract in order for you to move fast in order to do the exercise. So we've got to link this idea of less gas exchange and how that might mean that exercise is hard. So damage to the alveoli would mean less oxygen diffuses into the blood. That's the link between the damage to the alveoli and the gas exchange. Now we need to link less oxygen to exercise. So if there's less oxygen in the blood for respiration, there's less energy to release for muscle contraction, and exercise obviously relies on that. So that's why if you have less energy, exercise would be hard. So I've written my answer out with the steps that we just said, but I make sure, and this is something that people often don't do with these types of questions, if something is changing, so something is damaged or broken, and that will have an impact. Either you need to say something like, oh, if we need more oxygen for respiration to release more energy when we're doing exercise than normal. The same way if you're saying something's not happening, so less oxygen, less respiration, less energy. You'll notice in the mark scheme, it only says we need to say the word less once, and that gives you the indication that that's what you mean. But always get into a good habit of using those more or less words, really important in questions, so when you're explaining why something's happening. So if I've said less oxygen's in the blood, what does that mean? Less respiration. What does that mean? Less energy released. So just try and make sure we include those when we're explaining when there's a change and why that's the knock-on effects that that's causing.
Well done for making it to the end of this paper. I'm really, really proud of all of your hard work.